Alrighty, well, welcome to HIV, and we need to learn of it for Biochemistry 2, BCH 4053. So today we're going to be looking at HIV, its life cycle, and yeah, so let's get started. So what is HIV? HIV is an SS, let me make this a little bigger, too big, there. It is an ssRNA virus. Now what does that mean? That means its RNA is single-stranded, and that is what it puts into the cytoplasm. What does that mean? Well, when HIV, we'll draw it out here. When HIV reaches a cellular membrane, it needs to register if it can attack it, right? The way it does that is that on certain cells, particularly on the ones that HIV target, which are these T helper cells, which are part of your body's innate immune system, they have these things called CD4 plus receptors. So those CD4 receptors, the HIV has these glycoproteins. Now what are glycoproteins? You can think of them as proteins with little sugar molecules attached to them that allow the virus to bind to certain receptors, in this case the CD4. So HIV comes over and has these receptors. These receptors bind to these, the glycoproteins bind to the CD4 receptors. And when that happens, let's draw this out. This gets pulled in with the help of a co-receptor on this. And then this can either be the CCR4 or the CXC5. I'm not sure about that. But it has two co-receptors that it's able to bind to. And in fact, in some patients who were immune to HIV, they discovered that their receptors, the co-receptors, were mutated to the point where they weren't able to actually interact with anything, and therefore HIV wasn't able to bind. But when it does bind, it binds here. You're now going to have a little pocket where this is pulled in, the HIV. So viral load here. So little receptors binded to it. Pulls this in, and inside this HIV, you can see right here, it has this RNA enclosed in what is called a capsid. Now, a capsid is just a protein matrix that lines around the RNA and protects it until it enters the host's cytoplasm. You see the HIV is being pulled into the cell wall. Here's the cytoplasm, and here's the environment. Okay. After that, the capsid, the lipids of both are going to combine, and you're going to have the capsid containing the viral RNA now be able to enter the cellular membrane. I'll just think of this as the environment, and this is where it's out of the cell membrane. Here's what the nucleus. So after a while, the capsid is going to degrade, and you're going to have this mRNA get released into the cytoplasm of the cell. Now, also with that HIV inside the capsid are these little proteins and enzymes that allow for this mRNA to be first transcribed into DNA. Now this is RNA dependent DNA polymerase. So that's the first thing that happens. Now another function of RNase, who's also in there, maybe you heard him before, he can degrade this RNA. Here, I'll label this RNA and DNA. He degrades this so you're left with just DNA in here. Okay, perfect. So you have this viral DNA, or vDNA. This undergoes one more thing, which is DNA. I'll type this out. My handwriting's not that good. DNA dependent DNA polymerase. Now this allows the formation. Now remember we had the viral DNA here. This allows for a double-stranded DNA to come. This double-stranded DNA inside the nucleus, which it can enter into, you have little parts of the genome that it can integrate itself into. Now it's important to realize that the way it integrates itself is you have, let's say your set of whatever, and then you have your viral part here. This is going to replace this. So let's say if you were unfortunate enough and HIV came and switched places with your cancer blocking genes, you would have a runoff of cancer, which is why DNA viruses can cause cancer. And there are very many famous examples, or one major famous example. By the name of Henrietta. I believe in Henrietta Lacks, who was very unfortunate, who is now living on in the HeLa cell line. She died, unfortunately, but her family sued Johns Hopkins over that. Anyways, so we have our DNA, and this genome is now hijacked. Oh no. It's the viral part. Now, the host's own RNA polymerase is going to be able to translate this genome into proteins, and that will form a couple things. One, it will form the vmRNA again, which can be put back into the capsid. It will form the capsid proteins, which are these capsids. It's going to form the enzymes again, the viral RNA dependent DNA polymerase, the DNA dependent polymerase, and RNase, and let's just throw that here, and it'll form it'll form mRNA for this, and then just form the vRNA separately. So this can be put back into the formation here. So we'll have our little HIV back, and now this can travel outside the cell, and it'll also grow the glycoproteins. I'll grow these out here. Let's draw them here. So once there's enough here, the capsid can reach the end, and this will combine fortification with the mRNA and the enzymes. And this will reach the outer cell. Once that happens, this will be able to leave the cell. This is the environment. Here's the cytoplasm. Here's the capsid. This is gonna get out of here, and we're gonna have little HIVs running in the background. So that's how it spreads. So yeah, HIV, not fun to have, pretty interesting to learn about.